Morning guys, I apologize in advance for the background noise. Grandma and Nicola are at home and they can be pretty loud when they're together. So today I'm going to be wearing this beautiful denim dress which I got as a present from my parents and I absolutely love. As per usual, I'm going to pair it with some of my Ana Luisa jewelry. As you guys know, I've turned into such a huge fan of their pieces. And for my perfume, I'm going to wear the delicious modern classic Miss Dior. So here is the outfit, guys. The dress is like a beautiful, very deep blue and it's made of like stretch denim. And my parents bought it for me a couple of months ago. They sent it over by mail because of course they couldn't give it to me personally due to stupid COVID. And I love it and every time I wear it I think of them. Quite shockingly, I ranked number 3 my iconic Illumination quad, and why it's shocking is because of all the 3 quads that were released last year, most people tend to rank this one the lowest and always use adjectives like boring and meh and not quite what I would fall in love with when it comes to Pat McGrath, and here I am unable to shut up about this quad. The reason, let me tell you. Personally, I feel like this quad epitomizes the reason why I fell in love with Pat McGrath eyeshadows. I know that she can do colorful things well, I know that she can do super like sparkly things well, but what I think made me fall in love with her brand is the fact that when she does neutrals that you can literally drool over. And this is for me what this quad represents. Gorgeous neutrals. Neutrals that are not boring. Neutrals that are complex. Neutrals that just look stunning on the eye. So as someone who comes from, I never want to wear neutrals, they're boring, I found Pat McGrath and I fell in love with fucking neutrals. And this quad is a perfect representation of the reason. This quad is not the reason why I fell in love with neutral eyeshadows, because I already had many other eyeshadow palettes from her, but it is definitely a representation of what I love about her brand. Just making the most boring colors look complex and gorgeous. Going to shut up about it now and we're going to proceed to the look and a little bit more of story time. So just let me tell you, I'm going to use all the 4 eyeshadows in this quad for today's look, and I am going to pull out one additional shade, which is this shade Statuesque, which is just like a neutral taupe matte that I will put through my crease. But for the rest of the shades, this is going to go into my outer corners, lids, inner corner and a bit all over the lid, lower lash line. Now, to my utter surprise, you guys reacted very positively to my story time last time, to my nerdy story about what I did during my first internship at the lab. So today we're going to continue in chronological order, and I will tell you what happened when I went back to the lab to pursue my PhD studying Gene X. So when I started my PhD, it was still Gene X, because even though, just to recap from last time, because maybe some of you missed it. During my first internship at the lab, I discovered a mutation in a disease called myelodysplastic syndromes, which is a blood disorder. And I found out a new mutation in a new gene that had never been described before. So I wanted to go back to the lab to study the protein product from this gene. Just to recap, DNA is kind of like a blueprint, and one of the most important things that it produces is proteins which are encoded from genes in the DNA. And proteins are the workhorses of the cell. They just make shit happen around the cell. So we didn't really know what this protein does because it was annotated as a hypothetical protein, and we had absolutely no idea what its function is. And when you're someone like me who enjoys basic molecular biology, one of the things that thrills you the most about biology is finding out how shit works and what it does. And finding out what a protein does ranks up there amongst the most interesting things that one may discover in biology. So like I said last time, when I finished my internship, I still had to go do an internship in another lab, which I'll tell you about more one of the next times when we talk about my postdoc. But I had to basically take about a year off because I needed to go back to finish my study, do my second internship, so on and so forth. And in the meantime, they had sequenced the gene in multiple patients with this blood disorder, and they had found out that the frequency is about 25%. So at the time I went back, everyone was already preparing to publish this story. Now let me tell you, this is one of the interesting parts of this discovery and also a little bit of a glimpse of how science works a little bit. So like I said, it was a really big deal, so we were keeping it under wraps as much as we could. We were calling it Gene X during presentations. Nobody was allowed to talk about it outside the lab. You know, it was a real hush-hush situation. And of course we wanted to publish it in a really good journal. Now, I started in September, and in the month of December we were already almost done with like submitting our paper. So in blood disorders, the biggest meeting in the world is one that takes place in the US, and it's called ASH, which stands for American Society of Hematology. And this meeting always happens in December. It's a huge meeting. I've been to that meeting once. 20,000 people attend that meeting. If you want to go from one part of the conference building to another part, you literally may have to walk for half an hour. That's how big it is. And you have to like really schedule what you're going to see and when you're going to see it, because if you just go there and want to attend a few meetings, it's not going to work. There is so much going on in parallel, you need to be prepared. But that's like a total sidetrack, sorry. So, we heard that there was a late breaking abstract, which means that it's like something last minute, something super exciting, and it was announced right before the meeting that someone was going to present that they had found mutations in this gene as well. Now let me tell you, this other group discovered the gene independently of us, totally fair and square, and honestly it was not that surprising because it was just there. We just needed to up the resolution in order to be able to see it. So someone was going to find it eventually, and it just happened to be us and that group it was a group in France. So they were going to talk about their discovery. But what happened, unfortunately, is that when they announced that late-breaking abstract, some of the sharks in the hematology field, especially one guy, jumped on it, and he just immediately went to the lab and told his army of PhD students to start sequencing the gene in patients. The sad part of the story is that neither us nor that group in France were the first one to publish the mutations in this gene. It was that asshole who jumped on it and basically produced a paper in a month. So even though his paper was crappy and it wasn't published in a very high impact factor journal, the mutation, or like the announcement for the mutation and the gene, was out there, and it was not made by us or the French people. We and the French people published our papers almost back to back into very high impact journals. They published in the New England Journal of Medicine, we published in a journal called Nature Genetics. And it was it still didn't take away from us, because that asshole published in a much lower impact factor journal, and our papers are still the ones that are being cited as the ones who discovered the mutation. I think people in the field just knew who discovered these mutations, but at the time, me and my supervisor, the PhD student who was going to be the first author on the paper, were a little bit bummed out that we didn't get to announce to the world our discovery. So that's a little bit of a glimpse for you how science can work. It's not all, let's work together and solve all diseases. It's not like that. Scientists are very competitive, and the ones who make it to the top tiers are sometimes the biggest assholes. But anyway, let's go back to the story of my PhD and the fact that I wanted to find out what the function of this protein is. Something that we do in the lab very often when we want to study the function of a protein is we produce a recombinant protein. So basically we take that piece of DNA, we put it in a circular DNA, which is called a plasmid, which you can put into bacteria, and the bacteria are basically used as factories for multiplication of that circular DNA, which carries your protein of interest. So we did that, and we also plugged a little fluorescent tag in front of it. A fluorescent tag is a protein which is present actually in jellyfish. It's called GFP, green fluorescent protein, and we can plug this green fluorescent protein right in front of our gene of interest, produce multiple copies of that in bacteria, and then put that back into mammalian cells and see where it goes, because it emits green fluorescence, which you can see under the microscope. Which is one of the first things that you want to do, because when you find a new protein, one of the most important questions you have to ask is, where is this protein localized? Because it can tell you a lot about its function. So I remember the afternoon that I first went to the microscope to look at the localization of my protein. I went to the microscope and I saw it. I'm getting too excited here. And I saw it beautifully localizing specifically in the nucleus, in this sort of like little like beads in the nucleus, which probably signified that it associates with the parts of the chromosome which are actively produced into proteins in the cell. We call that euchromatin, which is the active part of the genome. I remember going to my boss and telling him that it's a nuclear protein, so it must have something to do with regulation of, you know, DNA or transcription, which is basically the process through which you produce RNA, because if you guys probably have heard about it in high school, the central dogma of molecular biology is DNA, RNA, protein, in that linear direction. It's not quite like that, but let's put it that way. So we thought it must have something to do with regulation of DNA. About the same time, and this is another, you know, thing that is so very interesting in science, another group, unbeknownst to us, was searching for the function of this protein, but they were coming from a completely different side of the story. They were using bioinformatic tools in order to predict a protein which might be able to perform a certain function, and I'm going to tell you a little bit more about that function because it's actually quite interesting. So, like I said, DNA is a little bit like a blueprint, and you have the 4 bases, A, C, T, and G, which form these sentences called genes, which can be translated into proteins. Now, what happens very interestingly, I'm going to start applying my lid shades, by the way. What happens very interestingly is that one of those bases, it's called cytosine, stands for C, can have an extra modification on it, and this extra modification is called methylation. And when you put this methylation on cytosine, and it's in a specific context on DNA, which contains a lot of C's and G's together, and we call those CPG islands, and those are very often present in the promoters in front of genes, and promoters are the thing that kind of stimulates the transcription of genes. When you put those CPGs and the methylation, so the extra modification on that C in front of genes, it means that these genes will be silenced. So when you have that CPG island and a lot of like methylation on the C's, it means that the gene will be silent. So the chromosome will tighten up, the DNA will not be accessible to all the factors that are needed to translate it into an RNA, which will eventually be turned into a protein. DNA methylation is a so-called epigenetic modification. Epigenetic because literally it means above genetics, but it is transmissible. So you can inherit this even though it's not part of the actual genome. What's very interesting about DNA methylation, it's called, this DNA modification, is that, for instance, something you may not be aware of, but all of you females have one of your X chromosomes methylated and inactivated, because as you guys know, men have only one X chromosome, females have 2 X chromosomes, but in order to not have 2 copies of everything that's in the X chromosomes, one of our X chromosomes needs to be inactivated, and this is the process that takes place. It's called X chromosome inactivation, and DNA methylation is what makes it happen. So the thing is, DNA methylation and the enzymes that perform it were known in the field for at least a decade, but there was huge debate on how to revert it. Clearly it was reverted because genes can be like switched on and off, but we didn't know how. So this group in the US was using bioinformatic prediction tools to find an enzyme which might be able to do that. And in that way they landed on one of the cousins of the gene that we were studying. Our gene has 2 cousins and they all can perform this very same function, because as it turned out, they are the enzymes that revert that DNA methylation. So they can go from silencing to activating. So these guys were not studying hematology, they were not studying mutations in patients, they were trying to find out what reverts DNA methylation, and that's how they ended up on our gene X. So in the end, I didn't have anything to do with the discovery of the function of this protein. It was this group in the United States. And I think they made a really remarkable discovery because they solved a decade-long debate in the field. I went on to show that mutations in patients deactivate the protein. We call that loss of function. So instead of going on the part of DNA that is silenced and activating it, it can't. It can go to that part of DNA, but it cannot activate it because the enzymatic function is disrupted. And that's what happened in patients, and that's what I was able to show. Another part of my PhD was showing that this, by the way, I'm applying a tiny bit of the brown now just here onto the outer part, and after I'm going to use the red shade in my waterline and the rest of my lower lash line. We found that mutations in this gene occur not only in myelodysplastic syndromes, but they also occur in patients with, for instance, acute myeloid leukemia. And what I showed was that mutations in this gene, when present in patients with acute myeloid leukemia, bring about the worst prognosis. So they are a poor prognostic factor, which has a lot of implications when it comes to the clinic. Yes, we don't have a treatment for it, but if we know that it's a poor prognostic factor, clinicians can decide to go a bit more aggressive with the therapies in order to achieve remission in patients. These are pretty much the main discoveries that I made during my PhD, and trust me, work at the lab, especially at the basic biology level, is very challenging. It's filled with a lot of disappointment and a lot of failure. So it may seem like I didn't really discover that much in the end, especially not the protein function, but keep in mind I was a PhD student, so basically I was an amateur, and on top of that, it was just it was a very challenging project. It turned out to be something really spectacularly new, which on the one hand was really cool, because it meant that we made a real contribution to hematology, but it also meant that people from epigenetic regulation, so the people who study epigenetic regulation jumped on this, people who are big in hematology also jumped on this, and our lab was not a very big one, you know. We didn't have multiple PhD students working on this project. It was just me and a technician. That's it. So we couldn't really compete with all the big labs, I have to say. That's another one of the reasons that I think it was unrealistic for me to expect that I'm going to make significant contributions or more significant contributions than the one that I did. I'm still very proud that we discovered the mutation. I think nobody can take that away from us. But further than that, in terms of the function of the protein, it was up to other people to find that out. Alright you guys, this is the final look I chose for a pretty simple neutral lip with this eyeshadow look. I could have gone for a red lipstick, but I've been wearing red a lot lately, so I thought I'd give it a little bit of a break. What I'm wearing on my lips is a combination of a lipstick and a gloss. The lipstick that I have on is the shade Fuel, which was part of the Naked Heat collection from Urban Decay, which is basically like a nudie peach color. It's really pretty. And over top of that I put one of my new Pat McGrath Opulust glosses. This one is in the shade Dreamscape. Oh my goodness, these are so gorgeous. I'm going to talk a little bit more about them during my Black Friday haul, because I have two more. I bought a little set from them. This is just one of the colors, but I don't even think a camera can capture how these shift in real life. This is stunning. So I just put a little bit of this mixed in with the lipstick. So I put on the back of my hand and then I work it into the lipstick. I don't like to apply a full-on layer of lip gloss over the lipstick because I feel like it moves the lipstick around, especially because Fuel is a cream lipstick. Now, number 3, Iconic Illumination. I will be forever loyal to this squad. I will never shut up about it, and I will never stop recommending it because I think it is just absolutely stunning. Every single shade in here is so beautiful. This shade is so stunning and so different. This shade literally makes your lids look wet. This is a gorgeous deepening shade. This is a stunning burgundy shade. I just will never shut up about this squad. If it's still around and you are thinking about getting it but you are unsure, if you love neutral eyeshadows, trust me, you will love this squad. Next week we're going to see each other for number 2, Subversive. It's going to be a very special episode. I'm not going to tell you why, because, well, first of all, it's going to be a special episode for a reason, and second of all, story time will also be something a little more special. So we will see each other next week. Thanks so much for watching, as usual. Don't forget to like and subscribe if you would like to see more of me. If you'd like to see all the videos that are in my Pat McGrath eyeshadow palette ranking list, I'm going to leave that up here for you so that you can check it out. Thanks so much. I'll see you next week for number 2 on my ranking. Like I said, that is the Subversive palette. Bye!